Welcome everyone to our Multiscale Neuroscience Journal Club. We are going to talk about the cerebellum. I picked one of the papers, Boston and Strick, but mostly I've just sort of put some diagrams together. And since I myself have not actually thought about the cerebellum in quite a while, I'm hoping everybody else will have interesting things to say about how. So basically, this paper is sort of like a lot of what and not a lot of how. So, but we'll get to that. But this also, let's just revisit the cerebellum in case a lot of you haven't, like me, haven't looked at it in a while. So yeah, it's the little brain, and that's where it is. And we've been looking at this diagram from Paul Chisek's paper, and so it grows out of the same segment as the thalamus, but it's for some reason not in this the diagram, this fate map kind of thing from this hypothetical vertebrate. But so yeah, here's the structure of it from kind of behind and slightly under, I believe, is this is the view of it. And if you slice through, you find something that sort of like the cerebral cortex, the cerebellar cortex, but to my eyes, there's something almost sort of plant-like about the way the structure, the shape of it reminds me of something plant-like for some reason. And so you have these deep cerebellar nuclei, the most famous of which is the dentate nucleus. I don't hear that much about the other deep cerebellar nuclei. Emboliform is one of them. So if you know anything about those other nuclei, Mac, for instance, please say something.
Yeah, fastigial and interpositus are the two other ones. Fastigial is involved in balance, so you probably don't think about it much if you're able to kind of keep yourself sorted. And interpositus is sort of motor related and a little bit less interesting for people interested in cognition, because the dentate is, the fascinating thing about the dentate is that it's expanded over evolutionary time. So it's bigger in monkeys than in rodents, and in humans than in monkeys, projects to the mediodorsal nucleus of the thalamus, which has then projections to the frontal cortex. And so there's lots of really interesting implications of its anatomy and function over phylogeny that get people really excited.
Cool. So that's, I don't know whether you can see the captions, but dentate is here. And so that is there. And so yeah, I was just saying that I just came across this book. I was looking into, right at the end of this PowerPoint, I was thinking, maybe it's a good time to talk about Mar Albus. And maybe a more accurate term is Mar Albus Ito, but Ito himself, Masao Ito, was influenced by David Marr's ideas. But apparently David Marr was looking at diagrams from this book, and I was looking at a recent historical review of the Mar Albus model of the cerebellum. And so I thought, well, let me have a look at this book. The diagrams are really beautiful. And a lot of modern diagrams appear to be redrawings or like tracings of like, for instance, this very diagram. I've seen papers that have basically traced it. So yeah, you'll see the famous groups of neurons, the sort of flat, the Purkinje cells, which you've probably seen Ramon y Cajal drawings of. The granule cells are in green. Granule neurons, parallel fibers in green. Not exactly green, but anyway. So we'll talk about this in a more schematic way. But I remember the first time I heard about the Purkinje cells when I just joined grad school. Dan Bullock, who would go on to be my advisor, talked about how they were so sort of tantalizing for a modeler because their this literal three-dimensional shape kind of reminded people of like, sort of cards that you put in like a motherboard or something. They're like, you have this big fan out structure, and then they're kind of in sequence like this, and then the parallel fibers from the granule cells run sort of perpendicular to them. But yeah, you can see them here, these parallel cells. We'll show in a schematic way what the circuit looks like. So yeah, we just talked about the deep cerebellar nuclei, dentate nucleus being the most well known or talked about. This is a sagittal view. So just I find nowadays, since I'm in an anatomy lab, I like to get some bearing about where these places are. Sometimes modelers just completely ignore that, but it's nice to know. So yeah, here's the classic circuit. that people have been looking at since the 60s. So this is so you have these afferent neurons. The mossy fibers come in from the pontine, the pontine nuclei, which is the main way that the cortex the cerebral cortex communicates with the cerebellum. You have these mossy fibers coming in, which synapse. I believe these are excitatory. Yeah, this plus here on the granule cells, and they in turn produce these long parallel.
Yeah, those are the granule cells there in orange. There, Yohan. The GOC is Golgi.
Golgi cells. Yeah, these are Golgi cells.
Yeah,
granule cells here. Yeah. So these produce the parallel fibers, and kind of sort of go across the sort of flat structure of the Purkinje cells, and the Purkinje cells then inhibit. Right, they're inhibitory. They inhibit the cells in the deep cerebellar nuclei, dendrite, for instance, and so that's the main output station. And there's this sort of interesting loop back where the synapses on the inferior olive, right? And this goes back up to the Purkinje cells. And people have sometimes sort of leave out this whole story, but one idea is that this serves as a kind of teaching signal. If we get back to the modeling stuff at the end, we can talk about that.
So they don't show it here, Yohan, but the deep cerebellar nuclear inputs back to the olive are inhibitory, but their projections out to, say, the thalamus or the red nucleus are excitatory. So this kind of is thought to close a loop and tell the olive to kind of diminish its activity, and you can see the kind of crazy influence that the olive's connections look more like the glomerulus in the kidney than they do any other neural connection that we come across, right? Where they're kind of like wrapping the entire dendritic tree. And they actually cause, I don't know if you're going to talk about this stuff later, but just to preface it, they cause what's called a complex spike, which is the release of calcium in these Purkinje cells that massively augments their responsiveness to the kinds of inputs that they'll get from other areas, makes them much more likely to spike and fire. The other thing that they don't really talk about in this paper, but is becoming a little more clear, is very important, is that there's another, just like in the cerebral cortex where we talk about, let's say, parvalbumin and somatostatin neurons, right? You could imagine this Golgi cell might be a little bit like a parvalbumin neuron where it's getting excitatory inputs but are also going to the target, right? So this guy here, and it might give you something like a feed forward inhibition. And then you might imagine that something that's looking at the dendrites or contact the dendrites might give you something more like a feedback inhibition. There's another cell type known as a Lugaro cell that actually acts like the VIP calretinin type neurons that inhibits both of those. And for reasons that are probably a bit selfish for a personal discussion with Yohan, these guys here have 5-HT3 receptors like mad. So they're really responsive to serotonin. So you can basically turn on, the inotropic version.
And is that excitatory or inhibitory?
We think it's excitatory, and we know that serotonin excites the cerebellum. So there's a lot of like transitivity going on where you find little bits of evidence and you have to try to guess what's happening. But there's a kind of leading hypothesis that serotonin increases excitability of these cells, which would be like disinhibiting now the whole circuit. So now you shut these guys down. You shut them down. Now the whole thing can run at a higher level than it would have beforehand. It's sort of a little bit like the striatum or the VIP neurons.
Right.
Anyway,
so what targets the Lugaro cells?
In terms of excitatory inputs?
That's I guess like any
great question. Yeah, so they've got a lot of serotonergic fibers from the pontomedullary reticular formation. I know about that for sure. I don't know about the rest. And this is one of those areas that is sort of like a forefront that is like vastly understudied in literature. If you could take 90% of the people doing hippocampal research and put them onto this stuff, I think we'd be in a better place in 10 years. But that's just my opinion, not the funding agency.
I can totally see. Yeah. We need the zoo of inhibitory neurons for the cerebellum, right? So cool. So I just put this out here. We'll kind of come back to this after we see what Boston and Strick are talking about. I thought this was a great diagram. What I do now is when I want to make a presentation is always Google Images, right? And I'm saying, okay, what's a really, really good diagram that I can actually cite and say, look, yeah. But I thought this was really, really nice, halfway between a kind of anatomical accuracy and something that a modeler would like to like work on. So yeah, this is why they're called parallel fibers, although there's kind of topological straightening that you need to do in order to get it to look like that. And so yeah, there's this kind of pooling in to the deep cerebellar nuclei, which I feel like there's not enough sort of commentary on that aspect of the whole story based on my understanding of the Mar Albus thing. Because if there's a fan in, then there's a dimensionality reduction, and you have to kind of say, well, what does that mean? And maybe someone who's looked at models of this lately will have some ideas about that. So this is hopefully you all recognize that on the left that's a rodent. But so just the circuitry from the deep cerebellar nuclei, where does it go? Mainly to ventrolateral thalamus and then motor cortex. But there are, as we'll see, other thalamic projections that can have an influence elsewhere. I'm not 100% sure about this. Does the cell, do they project to MD? Probably not, right?
In rodents, I don't think that if there is a projection, it's not supposed to be a very strong one. Yeah, the idea is supposed to be that becomes much more elaborate in primates and then humans.
But so it does exist in primates.
That's the story. Yeah, there's a Provosto and Somna sort of perspective-y review on it that kind of lays out a lot of the evidence.
Cool. So yeah, I just wanted to sort of point this out. This is the main kind of circuit that when people talk about when they talk about cerebellum as a motor structure. This is the going to cortex, and this is the cortex kind of reporting back to cerebellum. What that means, still up for grabs to a large extent. So this is the paper that I sent out, Boston and Strick. I knew that Strick was I just looked for Strick last week. I'm curious about sort of non-motor effects of cerebellum on thalamus and cortex. Kind of came up at the end, but basically what they've done is they've looked at this two sets of data basically about the disynaptic communication between basal ganglia and cerebellum in both directions, and looked at the possible implications of that in disorders and in normal functioning, but without any kind of real sort of explanation of how or why or what. But just saying by virtue of the fact that there are these connections, this will kind of maybe help explain some of the correlational effects and even some of the lesion effects. So the method is the classic old rabies virus method, which some of you who haven't looked at the method sections of these kinds of papers probably won't be aware, but so prior to the modern immunohistochemical kind of way of doing injections, like what our lab does, my lab does, is you could inject the rabies virus and it would be taken up and it would go sort of back, what's the term for it? Not antidromically, but the anatomical equivalent to that. Anterograde uptake would happen. And then depending on when you sacrifice the animal and look, you can like the amount of time it determines whether it's traveled one hop or two hops back. So you can infer from the number of days that you waited whether the cells that show the stain are monosynaptically communicating with an area or disynaptically. I think it starts to sort of fade off eventually, but I think you can do 2 or 3 days. But it may get really confusing after 3, what exactly you're looking at.
Johan, any idea how robust that estimate is of the retrograde transport, the transsynaptic portion in particular? Like,
I haven't looked at the numbers, and it's and nowadays most people don't use that method anymore. So Peter Strick is one of the few people continuing to use it. So maybe I'm sure there's others, but like with the rodents, there's all kinds of stuff you can do with all the fancy genetic methods, and immunohistochemistry is really magic. so like you can do all this amplification stuff and like triple labeling and all. I mean, it's amazing what you can do with immunohistochemistry. So And in those kinds of injections, you can do retrograde, anterograde, both, but they will stop. They'll only go one hop. So that's what people tend to use now, plus the optogenetic stuff, which is physiological, but you can infer based on timing how far something is. So yeah, the injections in putamen, which is part of the striatum, the more motor oriented part, receives input from one of the intralaminar nuclei of the thalamus. We looked at, I think we said there was the diagram we looked at last week, and which in turn receives input from the dentate nucleus. And in the reverse direction, the subthalamic nucleus, something that it seems like a small area that's not very famous, but I feel like it's sort of important. It's the only excitatory node in the basal ganglia circuit. So it excites pontine nuclei, and which in turn go to the cerebellar cortex. So they're just the only point of this
might be worth
what this means.
Sorry, it might be worth jumping in really quick just to kind of give a little bit of context. This population of neurons in the dentate nucleus that projects to the kind of intralaminar thalamus, which would then have the thalamus straddle connections to the putamen, for example. Another version of this would be the PF, right, which would go to caudate. These are present, but by far the minority. The vast majority of cerebellar output cells project to the core-like nuclei, which are the ones that project instead of to the striatum up into the cortex to the more the granular layers. So there'd be a massive population of them going to VL and then in primates to MD. So there's this pathway does exist. It's clearly there. There are other projections from say DN to the VTA, DN to the locus ceruleus, DN to a bunch of other structures that are kind of in that more diffuse category. But it's really important to know that they're the exception to the rule rather than the standard kind of plan for that structure. So I think it's just worth keeping that in mind.
In this pathway that you're describing right now, I mean, it really seems like it's almost identical to like the sensory pathway. So, I mean, up from at least from the thalamus, right? Like you have input from the cerebellum rather than the sensory periphery.
Yes. So if we're imagining a little cute diagram of a thalamus, right? Anterior nucleus, let's say medial dorsal, and then you have like the lateral and the ventral anterior, ventral lateral, and then maybe lateral, and then back here the pulvinar. Let's just say not a great example, but just to kind of give you the feel for it. If you were like the cuneate and the gracilis nucleus, right, which are coming up from the dorsal horn of the spinal cord, they'll come up and project into say lateral dorsal, and then they'll project on to an LP and then onto S1, right? And if you're LGN sitting back here, you'll get retinal input, right? And it'll project to LGN and onto V1, right? So the sensory pathways have this particular projection pattern, right? We call this like the kind of core-like nuclei, but interspersed with all of them are also ones that project right diffusely to the cerebral cortex, and we call those the matrix type. Right. This is like the Ted Jones terminology. In the anterior part of the thalamus, the anterior nucleus is weird, so let's just put that to the side because it's really connected into the hypothalamus, the mammillary bodies, as well as parts of the cingulate. But VA and VL are kind of receiving either cerebellar inputs or basal ganglia inputs. And the way I think about that is the cerebellum has an L and basal ganglia has an A, but that's not a very good acronym. I'd be kicked out of medical school for that shitty mnemonic. But so yeah, the VA receive these basal ganglia inputs, and the cerebellum project into the VL, and these guys are the ones that project in the sensory-like, if we call it quote unquote, LGN-like way to the core nuclei, whereas these VA VLs are the ones that project up relatively diffusely, and MD is a good old mix of both of those types. So it has a lot of the core type and a lot of the matrix type kind of all blended in together. And it's still a bit of a wild west in trying to work out exactly how many of the projections are coming from areas like the basal ganglia and the cerebellum. We know they both contact cells in the medial dorsal nucleus versus the amygdala, the nucleus accumbens. There's a bunch of different areas that project into MD as well as cortical region. So it's a bit of a mess. And so it's a bit harder to sort of like really delineate something and say, here's the general pattern. But in the sense that this pattern here is kind of the exception to the rule, whereas the more typical one is the projection that does that, like you were saying, like the sensory structure. Sorry if that's a bit of overkill, but it's worth putting some of these things into context.
Because this is also a bit of a tangent, but this is the best time to do it. So I spoke to Bacillus in my lab, who's pretty well versed in the thalamic anatomy. But I was asking him about the core versus matrix issue and basal ganglia output, and it turns out that it's not that well known how well the GPI selectively projects to only matrix versus core, and most thalamic regions have both core and matrix.
Both, yeah.
But interestingly, there's a medial-dorsal, lateral-medial kind of spectrum. So as you might expect, medial thalamic regions have more matrix-like projections, which are the ones that go diffusely to the upper layers. Whereas as you go more laterally, you can get a lot more, relatively speaking, more core projection. So it's possible that these within a given nucleus, they are still somewhat intermixed. But even there, you may have some gradients. So the relative amount of one versus the other is different. But you're saying that not that many people have looked at this. So that's interesting.
Yeah. The main evidence I know comes from a couple of Japanese groups that have done work in rodents and primates, but you're right, it's not again, we could kind of broadcast this to the NIH and say we should start funding cerebellar and basal ganglia circuitry research rather than hippocampus.
Yeah.
Because we just don't know enough about it.
So this just seems like this is the kind of circuit where you can do like mental rehearsal in, right? The cerebellar circuit.
I like you. I mean, you're going exactly to where they want to go in the paper, but they don't really flesh it out. But I just want to say one thing. When you said that this sensory circuit thing, there's an interesting way to like do that analogy. So the retina, the LGN, the synapses that the retina forms on the LGN are these huge, very large, I forget they're called triads. They're very large and they're very distinctive synapses when you look at them under the electron microscope. Interestingly enough, the amygdala projections to MD are like that. So Helen told me this one day just in the lab, and I immediately said, so the amygdala is like the emotional retina. And so it's like, so you can, meaning that there's certain, like for a given thalamic region, if you do find like a certain kind of very, very strong input, you can potentially make some sort of analogy about how relatively important that is in terms of what that thalamic region is broadcasting to the rest of the body. But in this particular case, the thing that kind of why I picked the cerebellum in the first place was, what does the cerebellum need to say to these cognitive basal ganglia loops? It's like, so you get this copy of this, and then why back to the putamen, right? So it's like they've said there is a connection, as we'll see, but what it means is tricky from our standard way of thinking about all this. So just to remind everybody about what the basal ganglia circuit looks like, there's always some like sort of lack of clarity I find in diagrams, like about whether GP projects back to STN and whether the GP projects back to the striatum. I'm not clear on this, and I haven't looked in a while. I see both kinds of present a lot, but yeah. So and then also
interesting link, Yohan. That projection you just mentioned, the ArchiPalatal Pathway, the one that goes from the GP back upstream to striatum and STN. The synapses there look a little bit like the inferior olives enwrapping of the Purkinje neurons dendrites. They kind of surround them in this kind of funny, looks like someone's like with a big hand has like grabbed a tennis ball and sort of like smushed it, versus other ones where it's like a tiny synapse onto a bouton or something. And that's a drastically understudied circuit.
So in striatum and STN?
Yeah. So specifically these guys here, it's called the ArchiPalatal Pathway.
Okay.
And I saw a talk by John Paul Bolam years ago at a movement disorders conference that basically did juxtacellular labeling in these things. So they would like, work them out electrophysiologically, and then the animal would be cold, and then they would be able to put dye into them so they could find the projections of these neurons afterwards. And they would come back up here, and there would be all these crazy tendrils going everywhere. But as soon as they found a STN cell, they would just wrap it like they were kind of covering it like in a ball of yarn. And so the idea was that they're exerting a ton of inhibitory control over ongoing circuits, so they can sort of blanket everything off. That was the functional kind of guess, but it's still massively understudied compared to the rest of the circuit. Anyway, I just thought that was a cool little nerdy aside.
Look into that. So I guess this is also fairly well known, but if you look at the striatum, it's sort of like a squished striatum, but it has this continuum in terms of which cortical areas it receives input from, and also which dopaminergic populations it projects to and receives. There's a kind of a spiraling structure. Suzanne Haber made that sort of famous. And that's something to keep in mind, because sometimes when you see these circuit diagrams, they're almost like a conceptual parasagittal slice in a sense. But you have to kind of think topographically about it. So yeah, the reason I showed that was so that because that makes this a little easier to sort of parse, which is from the Boston and Strick paper, where they're talking about the way in which different cortical areas communicate with striatum and cerebellum. That's the dentator. Yeah. So the interesting thing about the basal ganglia is that the striatum is it receives the cortical input from like the whole brain, basically the whole cortex, but it only sends output to frontal thalamic areas. So that's a kind of partial loop. We talk about cortico-basal ganglia loops. That's really only true for the prefrontal and motor cortex, and you can kind of see that reflected in the fact that sensory areas other than sensory areas are kind of completely absent from these GPI maps. Right, and is that true of cerebellum? I'm not clear on.
Yeah, so if we cast our minds back to that little thalamic diagram, they call it the ventral tier of the thalamus, which is typically VA, VL, and MD. And so they're the ones that are projecting to, in VA and VL's case, it's like motor, premotor, supplementary motor kind of areas, and then MD is kind of frontal more generally. I think there are some bit of rule breaking in primates, I think, such that you can have projections from MD that do extend further than motor frontal and can go to the parietal lobe and things like that. So the rules get softened a little bit, but the general principles, pretty good one to start with.
So in other words, it's fairly consistent the way that both basal ganglia and cerebellum interact with the cortex. They're talking to the same cortical areas, more or less.
Yohan, there's another thing here that I was surprised to actually find missing from this paper and from a lot of these diagrams. You mentioned, well, I was thinking when you were talking about how most of these connections are precentral, and the same thing's true for the subthalamic nucleus for cortico-subthalamic connections. They're almost entirely precentral.
Okay.
And then another thing I was thinking of, you mentioned Suzanne Haber's work on a lot of those terminal topographies. I actually think it's easier to see why those simple wiring diagrams are, in a lot of cases, not useful when you look at the explicit reconstruction work from like Kita in mice and Martin
the
Parent.
Haynes paper in monkeys is awesome as well.
Right, and Haynes Haber, right? That's one. But I actually think that even more useful than that is Martin Parent, 2018 Coude et al. I think that one's actually more useful. And why that came up is we're talking about red nucleus being receiving afferents from is it deep cerebellar neurons, right? Turns out that corticosubthalamic hyperdirect neurons from motor cortex, a lot of those collaterals actually perforate the subthalamic nucleus and go to the red nucleus. So there's sort of like a mini loop there, which I find really interesting.
Can you share that paper, Clayton?
Yeah, absolutely.
Yeah, that'd be cool. Actually.
Suzanne's work is brilliant. The Haynes paper, I was gonna bring that up as one to talk about. I don't know if did you find it in your travels, Yohan?
Say it again. What?
The Haynes and Haber paper. It's a J Neuro paper from probably going on 2013. There you go. Yeah.
Familiar, but I don't remember if
I think they, from memory, they did something like viral tracing of different cortical areas to work out which parts of the frontal lobes like projected into the subthalamic nucleus. and you can see this like really beautiful pattern of projections. And at one end, I think the ventral end, you'll have to correct me if I'm wrong, Clayton, the projections start to bleed over into the lateral hypothalamus. So rather than contacting the STN directly, they're actually contacting areas of the hypothalamus. And that's when, if you like listen to like Louis Puelas, he says that the STN is actually probably better described as part of the extended hypothalamus than it is part of the basal ganglia system. And it's just been kind of like roped into the basal ganglia because it has really strong projections to the globus pallidus. And, you know, functionally, it was a really important area for deep brain stimulation surgery, but it blurs. It's a little bit like the extended amygdala, right? Like some of these things, they have a name and we think, okay, this is a thing, but really part of what that thing is doing is sort of spreading out connections to lots of different areas. And so where you draw the lines can be really crucial.
Yes,
I really like that story. I think that the clearest takeaway from this, though, is basically dismantling the tripartite hypothesis of the STN. Because all the terminal regions are strongly overlapping. You can't really say, this third does this middle section does this, and this.
You know,
Okay.
Like it doesn't work like that.
Smear.
This is good. Yeah, this I remember this very clearly. Yeah.
And then you layer in this other thing, which is STN neurons. Their dendritic field actually extends. You know, in mice, it's like the entire extent of the STN. In primates, it's like a third, and it's probably comparable in humans. So you've got these terminal fields, but then you've also got these dendritic arbors that extend a third of the nucleus. It's hard to really say that there is a functional region. You know, this is limbic, this is association, this is motor. It doesn't work like that, unfortunately.
Right. They do mention sectors of the STN in this paper, actually. Right? So yeah, the sort of subtext is that they're saying something new about basal ganglia and cerebellum, but which maybe sort of sociologically people have been like that. They kind of, people who are interested in the basal ganglia on one end and then cerebellum people on the other. But I don't think that anyone thought that they were completely unrelated, except when people made claims about different time scales, like basal ganglia learning being on a slower time scale than the fast cerebellum learning. That's the closest I come. But I suppose no one has really proposed this integrated modeling of both. So at least I haven't come across any. So yeah, they're talking about these two pathways going one direction to the other, and they say it's an interconnected network. But you know, that's sort of obvious. So this is the sort of thing that they sort of go through once they sort of review what's basically in that diagram, is that things that we typically associate with basal ganglia disorders also seem to implicate the cerebellum. So in case of Tourette's, hyperactivity in STN may propagate to the cerebellar cortex and contribute to tic generation. So this was interesting. And this I thought was quite interesting. I did not know this, that resting tremor, one of the, we all know this, cardinal symptoms of Parkinson's, is optimally treated with surgical interventions or DBS in thalamic regions receiving input from the cerebellum. So that's specific. Interventions to the thalamic region that receive input from the basal ganglia are not as effective. So I haven't, followed those citations down, but can anyone think of, like, maybe is it the case that it isn't as clear cut? Because could that be one kind of issue there? But I thought this quite interesting. I mean, I wasn't aware of that, but maybe they're not quite as segregated even in the thalamus. So, but also, which
it doesn't come up a lot, Yohan. Oh, sorry. Go ahead. No, go ahead.
I was going to ask which thalamic regions receive only cerebellum and no basal.
I think it's a blend in all of them, but it's all about, like Bas was saying, it's a kind of proportionality. So VA is supposed to be more basal ganglia recipient, and VL is supposed to be more cerebellar recipient, and then MD kind of get both. This is the kind of classic story. But maybe just something clinically that'll be a bit useful here. So I remember when I was taught about Parkinson's in med school, it kind of got looped in together, and everyone's like, hey, yeah, Parkinson's disease, and that comes with tremor and stiffness and slowness and a mask-like face and responsiveness to levodopa and things like that. But the more I learned about it during my PhD, the better way to think about it is the better way to think about it is as a syndrome rather than as a disease, because there's multiple different causes of Parkinsonism, and Parkinsonism isn't the same for every person. And so having the stiffness and slowness is sort of one of these characteristic features, but the people that typically have resting tremor are very different group than the people that end up having sleep disturbance and cognitive impairments and hallucinations and other kinds of issues. And so they typically put them into two major categories. There's more than that in terms of clustering, but the kind of two major categories are tremor dominant, which they have a tremor that comes on and it basically is very frustrating for them, but it really doesn't impact much of the rest of their life, versus what they call the akinetic rigid subtype, which are the ones that are much more impaired motorically and have a lot more problems with cognition and sleep and mood and such. And so if you put all of them into the same category, you kind of make this mistake of the average looking nothing like either of the subgroups. And I think in some of these cases, right, with DBS, I mean, people that are experts can say more than I probably can, but the STN DBS works better for some folks and not others. And again, the VIM DBS can work better for some folks and others. And there's a lot of idiosyncrasies and individual differences in this field that often kind of just get papered over.
Really good, Mac. The only sort of anecdote to add to that is we are really, really careful when we talk about the effectiveness of DBS. We say it's effective for the treatment of motor symptoms of Parkinson's, because really Parkinson's is not a strictly movement disorder. It's also a cognitive disorder. And in fact, DBS can actually exacerbate cognitive symptoms of Parkinson's.
And affective symptoms too, right?
Absolutely.
I still remember a patient I saw doing my PhD being so excited to have DBS because he had some really bad movement problems. And then he came back in and he was just teary the whole time. He just could not stop crying. And his wife was like, "What's going on?" He's like, he used to be the most stoic man she'd ever met. And now all of a sudden he was just, breaking out in tears at every little thing that, every time a leaf would drop off the tree. And they ended up realizing that they'd put the lead in to the STN a little, they'd been stimulating too ventral, I guess was their hypothesis. And so they did a different protocol in terms of what they were stimulating along the lead in the STN, and they ended up resolving his affective disturbance and improving his motor, which I thought was a really interesting example.
So what you said right there, that's all you need to know to understand why industry has moved so strongly toward directional leads. It's the only thing you need to know to understand why that technology was developed.
Oh, cool.
Does it work? I don't know. I mean, it works. The problem is now neurologists have to take 10 times as long to find the right set of contacts to use. But anyway.
So yeah, they look at a few different disorders, and then they look at basically the correlations and things like that in normal function. So they found reward-related activity in granule cells. They found that activation in the anterior cerebellar cortex correlates with reward and reward prediction errors. Okay, not that surprising, I suppose. So basically, there are correlations. What they do is sort of less clear. Overall communication between cerebellum and basal ganglia could be essential to linking sensory motor adaptation to reinforcement mechanism. So I love how in review papers people get away with this is really, really a whole lot of information packed into this one sentence that you could spend a whole lecture on just explaining this one sentence. But what does this mean? So they have this diagram, which is all right, I suppose. I sometimes like overly simplistic theoretical ideas just as a tool to sometimes forcing something very, very simple onto a structure makes you sort of think, well, how could it be that something as simple as unsupervised learning is done by the whole cerebral cortex? Can that be right? And how? But yeah, what do these words mean? When you say unsupervised learning is happening here, or model-based control, model-free exploration, how clearly do we know what all of this exact model-free and model-based? I feel is a slightly, I don't know, odd way of thinking. There's a model-based and model-free statistics make some sense to me, but I don't know exactly if that's what they mean. But I see this a lot, and but yeah. So basically, fleshing out a lot of this is part of the job of modeling. And what I find is interesting is that like you can be an experimentalist and like published pretty broad functional speculation. And when you make a model, the modeler is often accused of that the model is too unconstrained, right? But how is that any worse than just a bunch of words? This always like irritates me about complaints about modeling. It's like, I agree that we have a lot of free parameters, but a proof of concept is always a good thing. And in my experience making a model, you always have to actually add some additional assumptions, or sometimes go back to the literature and say, well, if I do just this, it won't actually work unless I add this other thing. And then you say, oh, well, actually there's something in the literature plausibly that could do that job. And but yeah, like the hand wavy stuff is still really popular. Yeah, so in the early stages of sequence learning, activations have been observed in the associative territory of the striatum, STN, as well as cerebellar cortex. So this is stuff that I think is really cool to think about, these stages of learning, or like a part of the brain that's temporarily seems to be more involved and then sort of passes out of being needed. The sites of activation, the basal ganglia and cerebellum, are likely to be anatomically interconnected. Yes. And these interconnections probably provide the substrate for the observation that functional connectivity between basal ganglia and the cerebellum is enhanced early in learning. So why might that be? So yeah, it's like you don't quite know what's happening, and you're like learning is turned on everywhere. I suppose that's one way to think about it. But if like one of the ways people have thought about basal ganglia and cerebellum is that the sort of rough sketch of some sort of voluntary motor activity, which is relatively like above 100 milliseconds, 200 millisecond kind of time scale, involves the basal ganglia, and the finer adjustments on like the below that kind of time scale is done by the cerebellum. But maybe that's too much of a sort of sequential way of thinking. It may be that, in fact, the reason I've been thinking about this is I saw a paper from MIT, one of those mouse people, Japanese person at MIT, I'm blanking on the name, famous Nobel laureate. But anyway, so basically they found that instead of this, you know, the story about hippocampal memory transfer. What? Tonegawa's lab, yeah. So this paper from Tonegawa, one of those short science papers but it was a cool idea, at least, even if it's not completely right. But it was in mice, and they were testing this idea about memory transfer. In you know, that whole story that initially there's memory requires the hippocampus, and then through consolidation over a period of multiple days, the prefrontal cortex takes over the memory representation. So what they found, I forget the exact way that they did this, but is that rather than like a sequential story where first there's something happening in hippocampus and then the prefrontal cortex receives a transfer, what they found was that there was learning happening all along in both areas that was somehow being masked by, or like actively suppressed. And I just sort of really thought this was a cool idea, that what can initially seem like sequential learning phenomena, or like a kind of relay, a sort of functional relay, like where first this particular brain area does something and then transfers it over to somewhere else, is sort of more like a kind of difference in learning rates plus inhibition. So it's like, yes, all of you can learn at the same time, but the priority for which part of it gets access to behavioral control changes over time. And that's, I think, an interesting way to revisit some of these lesion studies and things like that. So it's like, another
James Houck has a nice way of putting this, Yohan. He thinks we should think of parts of the brain as forcing functions rather than as sort of independent units. So the cerebellum, by being connected to the basal ganglia and the cortex, let's say, it forces them to deal with the outputs of its computation. And in the same way, the basal ganglia, by being connected to the cerebellum and the thalamus and the cortex, forces them to deal with its outputs. And so by forcing them together, somehow the pressure that they each put on one another leads to an emergent kind of, operating mode that allows you to let's say learn a particular pattern or something like that. And early on in learning, maybe one of the forces has more of an influence on this, learning the accurate pattern. And then later on, once it's been learned and another part of the system can take it over, now its force can actually be to create variability that, let's say riffs on it. And so like, think of a really great jazz musician or something. Early on as they learn, they have to work out how to put their fingers on the keys. But once they've got that down, they don't have to worry about learning that anymore. Now they can think about, how do I play, how do I recover from that note, or how do I play that note on top of that chord rather than that chord or something. And they can like use the system in a different way. I find that really compelling as a way to think about all this.
Send me that paper, but also this fits in nicely with another hippocampus prefrontal cortex idea that I read it was part of a modeling paper, I believe Krasny et al., Krasne, and they were reviewing a whole lot of data. Quite a good, as a review paper, it was great actually. Modeling also was cool. And they were looking at, again, this hippocampal transfer consolidation. And they pointed out that there was a difference between lesioning CA1 versus reversibly cooling it. And they found that, and I think this is a well-known observation, that you can, like if you lesion the hippocampus before learning, the prefrontal cortex can actually subserve the same type of learning. But what's crazy is that you can if you reversibly cool the hippocampus, this doesn't happen. Or at least that was what the paper suggested. So it's sort of like the axons merely being intact, even if they're not actually firing, can somehow serve as a sort of break on the prefrontal cortex taking over. So I thought it was quite interesting. So obviously that is not necessarily a natural way to regulate the prefrontal cortex, but you could imagine that there's some sort of active inhibitory process that needs to kind of be turned off for the natural transfer to happen. So that fits in with what you were just saying, Max. That's kind of, so yeah, when I see these kinds of stories about transfer or stages of learning, that comes to mind, that maybe it's less of a transfer than unmasking. Which is part of this whole theme that I've been very interested in, right? Like thinking about all the different kinds of things that inhibition can do that we kind of maybe don't give inhibition enough credit for.
I have a question, Yohan. How
Yes.
can you, this is a very poorly formed thought, just warning. How might, so there are lots of decorticated mouse maze running experiments. And there's sort of a hypothesis that basal ganglia has some sort of like vestigial, I guess, generalized processing capability and learning capability. And I'm curious, if you were to remove the neocortex from these models, how would, I guess, the functional roles or I don't know, the discussion that you've been having, how might that be?
The model.
Right.
Like moving it from the model would depend on whether the model already had thalamic projections to striatum. Sometimes modelers leave them out, right? Because they, like, well, whatever. but we know they're there, and so the thing is that a modeler generally does not want to put redundant connections in because it just adds flops, right? and yet, especially for learning purposes, like if you're multiplying more and more matrices, it really adds up, right? So in terms of the time it takes to run the simulation. So, that's the answer, right? Like that, if you put some redundancy in that we know is there, anatomically we know that, right? That would give you at least something. Like presumably, like I've been saying this a lot, I use this term, what is all this cortical real estate for? So if there's something similar in thalamus and thalamus goes to this where there's a whole lot more space, representational space in cortex, right? Presumably that means that there's sort of sophisticated fine differences that only the cortex can really handle or hold on to for a long time. Whereas the thalamus has rough and ready versions of the very same thing that you can continue to use for signaling and learning. And then the fact that you have these, wonderfully large dendritic trees full of spines in the striatum, and the cerebellum presumably is also intact, means that maybe it'll take longer, or maybe it'll require a slightly different learning strategy, but in some sense, the information from the outside world is still present. So that would be how I would think about it, right? Like that you just make sure that there's redundancy in whichever stage is doing learning.
There's an alternative. Oh, sorry, go ahead, Glenn. You finished the thought.
Oh, no, please. Oh,
I was just going to say there's an alternative issue at play here as well. And again, I'm going to sound like a broken record here, but this is a fascinating circuit, the thalamus straddle circuit, and we just don't know almost anything about how it is wired up and its functional implications. There's some really cool recent work coming out showing that it's incredibly crucial for learning. There was actually a paper from a group in the US in Baltimore that that showed that you could basically lesion the motor cortex in a rodent, and you could cause problems with the striatum as well. But if you mess with the thalamus striatal projections during learning of a new skill, you would absolutely completely remove the capacity for the animal to pick it up. But here's where the rubber hits the road. We don't know enough about the projections from the thalamus to the striatum and their specific projection patterns. In fact, there's one tantalizing line of research that shows that they don't actually project that strongly to the spiny projection neurons, which are the ones that receive the cortical inputs, right, from the layer 5 cells, but instead project to the tonic cholinergic interneurons, which are inhibitory and have this weird rebound bursting that we were talking about last week. And so putting that puzzle together as a modeler is a challenge in and of itself. So just to throw in the connection as like oh, well, it's a redundant one is kind of like really challenging. So I just wanted to kind of highlight just how hard it is to do these things, right? Because we just don't know yet.
Are you saying that the thalamic projections to striatum do not synapse on this medium spiny projection?
No, I'm not being that forceful. What I'm saying is there's strong evidence that they have major projections to the cholinergic interneurons.
The TANs,
and some people, the TANs, and some people are of the opinion that that is their predominant functional role in the striatum, is to mess with the firing of the TANs, the timing and the likelihood of the firing of the TAN neurons at a particular point in time, which will have a huge impact over whichever part of the circuit those TANs are in. But it's not going to be as simple as learning some plastic change between I made some motor, action, and this is the likely reward. Again, it's dragging us away from this notion from the Alexander DeLong and Strick model of like a gating loop that releases a plan that then executes that plan when it's time to execute it. This isn't what the topology of the projections kind of naturally suggests when you start to unpack some of the detail. And what the algorithm actually is a massive open question. And I've speculated wildly about that, but other people have as well.
So that's the part of the story that I'm a little more familiar with, is sort of the concept that that at least with respect to the STN, is that it sort of gates conflict between competing action plans, competing motor plans, and essentially cortical input to the STN essentially establishes for the entire basal ganglia which motor plan is going to be released. But I mean, obviously that's all speculative. In fact, I found that paper, I don't know, Hold Your Horses. I don't know if that title is familiar to you.
Yeah, the Michael Frank one.
Yeah, I found that paper very speculative, but it's a very fascinating concept, and it sort of explains when and why the cortex interacts with basal ganglia. Yeah,
Michael's awesome, and he came from Randall O'Reilly's lab, who's also fantastic, and they're both really brilliant systems level thinkers.
Fascinating.
The model of the basal ganglia is like a little bit Alexander DeLong and Stricky, based on some of the things that I've learned of late, and that it becomes a major question. If you update one aspect of that model, let's say the thalamic cells that the basal ganglia are targeting, rather than having them directly come back and drive the circuit that was opening the channel, but rather have them come up and diffusely innervate the apical dendrites of those cells, which are now going to have a weird complex nonlinear relationship with whether or not that cortical cell fires, not just that cell, but all of the neighbors' cells as well. What does that do to the model? That's a massive open question that could lead in tons of different directions. You could show that it generally does the same thing, or you could show that it has really important subtleties that have led us down the wrong path towards thinking about the sort of simple function of the basal ganglia circuit. And this is where modeling is, I think, really well set up to work. And you just need time and effort and energy to be put into this to kind of figure out where the lines can be drawn and what the implications are, and what happens if we throw in this circuit or that circuit or refine this one just a little bit. There's a lot of work to be done here, man. It's as I keep getting told by my postdocs, modeling is not a trivial exercise. You've got to really sit and think long and hard about all the assumptions before diving into it.
Would you say 10% of the hippocampus people?
No. 10% can stay in the hippocampus. The rest get distributed out to all the other subcortical circuits.
I love that, but then you have all that competition.
So I think this point about the complexity of the known connections is well taken, but you could say that the reason people thought the basal ganglia was involved in action selection, and that is sort of like a consequence of the simplicity of the task structure. So there may be situations where you haven't actually given the animal all that many things to do within a particular category, right? So you're doing some two wildly different things. And so depending on how coarse-grained the multiple possible options are, it may look more like a basal ganglia centric choice versus where I have to choose between pressing key A versus key B versus key C, which are all very close to each other, which perhaps requires much more cortical input, or especially when there's a lot of detailed information required to settle the difference. So, and here's where you're right, the modeling thing helps, but you have to think about what the consequences of coarse graining would be. So what I mean by that is, if you say, instead of thalamus going to this sort of generic point cortex neuron, I instead replace it A with some sort of microcircuit, and B include this fan out, fan in business where matrix goes up, spreads out, and then it has to do something and then percolate and then come back to these deep cells, which then report back to basal ganglia, but also off to the races to premotor, motor, etc. Now, when you get rid of that, right? When you say that all of the different possible ways that I can reach the deep cortical layers are going to be considered equivalent, as you can do with the model, which is to say that a class of different possible actions from the perspective of the rich representation in cortex are going to be folded into one. Then it may not be possible to distinguish between basal ganglia is doing the competition and cortex is just listening versus cortex is doing competition. So it's like sort of in Paul Chisek's work, this comes up a little bit, right? That ultimately the whole circuit is doing something that is not quite attention and not quite decision making, and sort of both of this. Like it looks a bit like both. So it's like, I like to think of it as in terms of disambiguation, like the amount of information required to disambiguate between different courses of action. Sometimes it may not be a whole lot, and sometimes there may be a lot more. So rather than say that, don't make this simplistic model, it's like, see to what extent you can make a more complex model and then coarse grain it, and it'll look just like the simplistic model. So think of what you did, the Strick and who, the DeLong, was that what you said?
Alexander DeLong and Strick is the classic paper from when I think I was 4 years old. Yep.
So what you could do is imagine, like as a modeling hypothesis, that is like one of the modes of the basal ganglia, rather than a wrong conception, and not just modes of the basal ganglia. You have to say, link the task to the circuit and say that
in that task context.
When the task is particularly simple and involves pretty disparate, like maybe appetitive versus aversive, or like approach versus avoid. In those situations, I would actually expect the incumbents to be pretty much casting the vote between one or the other, with the right? So.
And where there's not that much uncertainty in the options as well. I like it when it's binary choice. It doesn't really matter if you've got variability. It's like, if you're not doing one, then you're doing the other. So it ends up just being a binary switch. So it's as if it's opening the channel for one or the other. So I like that idea, that you've got to really think carefully about what challenge you're trying to understand and what the computational degrees of freedom are in that choice before you can work out what circuit's doing. I like that. Right.
So the flip side of that would be that in easy situations, the fact that some particular, like when I say easy, I mean that there aren't that many countervailing pieces of information that make competition in the cortical representation complicated. Then the correlations even between what you sort of gate in the upper layers and what eventually happens is like kind of, it's very clear. But if for instance, the cortex is having to deal with multiple pieces of cortical information that are communicating with each other, and so while that sort of deliberative process is happening, you could imagine that they're recruiting a fair number of inhibitory interneurons to say, wait. We know that you want to eat, but it's not clear which of these things you should eat first. We know that apparently there is a poison, there's a spy, some of these things might be poisoned. So in that situation, I would expect basal ganglia has not that ability to make such discriminations. So it's like some sort of inhibitory. So it's almost like, and I think there's actually data that are approaching this kind of thing, which is the amount of clutter, variance, task difficulty, etc., will do things like slow down the urgency of making a decision and recruit some inhibition, for instance. And so that is where you can really start to say, not is it the cortex or the basal ganglia, which I think is not a particularly interesting question, but relatively speaking, which of these areas is contributing to a particular type of disambiguation? That's how I would see it. Or like the
I like
type
that
of
way to think about it.
You know, like exactly. That's the simplest way to put it. What type of competition is it that each of these structures is mediating locally? Because we know that the inhibition is happening locally. So you can say you can constantly say no, it's always at the network level, and I agree. Okay, but which inhibition is like really making the casting vote at the end? That you can actually ask about. Task structure and which sorts of inhibition is mandatory.
So you've got something like the task complexity here. Here's a complex task. Here's an easy, and then it's like easy down here. And then you've also got how familiar you are with the task. Let's say you're like, it's a novel task over here, and this is like an extremely well-learned task. And then you've got something like, which circuit are you using in terms of like the basal ganglia to cerebellum balance or something. You could just imagine different trajectories through this space, like leading you into, okay, the task got complex, quick, use some of your basal ganglia, but then you learned really, really well. So you can use the cerebellum, but wait, now there's a novel element you didn't see before. And you can imagine moving around that space interestingly with the different constraints of the circuit. Yes. I like thinking like that. That's good.
Right, exactly. So and the way, and this is why, my favorite thing, right, is inhibition. Like what are the ways in which inhibition can be recruited to bias a competition, shut down the dendritic tree of one group of cells, and those are the sort of the ways to sort of tip the scales in a competition, or slow down. Like, this is the other thing, right? This idea of slowing down the competition to maybe pick up on weaker signals that take a longer time to stabilize, for instance. So those two things might fit into this story, this disambiguation story.
Yeah. Not to mention neuromodulators, Yohan.
Right, indeed. All right. So in fact, there was this one interesting thing that they pointed out about the subthalamic nucleus. So yeah, they point out that all of us have often interpreted the STN as some sort of stop signal, because a lot of those tasks, including in monkeys, I believe, that involve one of those go, no-go tasks, which is different from the go, no-go circuit, where you give the animal a cue to say, yes, you can go and get the reward now. And then there's another cue saying, no, now you can't do it anymore. Those tasks apparently require the STN, without the STN, it's much harder for the animal to do that kind of thing, which fits actually quite nicely with what Clayton was saying about conflict. So I was thinking about this, that it was a different paper actually, where they were looking at STN in terms of movement initiation. And when I looked at that, I was like, well, isn't that kind of, initially that's sort of counter to the standard stopping story, the hyperdirect stopping story. And I thought about it a little bit, and I've been thinking about this, that we when we look simplistically at the notion of starting and stopping, we tend to think of an inhibitory signal as necessarily stopping and an excitatory one is starting. But that is way too simple, because when you've got multiple things on the burner, the starting of something new and the stopping of something else are bolted together. You have to do both at the same time. And that's actually a really subtle balance, right? And I love catching myself doing things that I do not know how to model, right? Like when you're in the kitchen and you drop something and you kind of reach and grab it, as long as it's not a knife. If it's a knife, you know that you're supposed to just let it fall. And like, there are all these little signals that we use to interrupt what you're currently doing to participate in some other action, versus decide that you cannot actually make it to that other action, and you have to do the opposite thing, which is sort of get out of the way or something like that. So this point about disambiguating actions, or like, is a very nice thing, because it shows that seemingly opposed functions, as simple as starting and stopping, can involve like just two sides of the same coin, and in this case, the SDN. So it's something that I really want to kind of look into for some other projects.
There's another sort of very closely linked dynamic to this, and that's that, and this is coming directly from DBS studies, that DBS can actually induce a bias of speed over accuracy. And how that relates back to this, initiation versus, rapid stopping, is also very interesting. And it sort of links DBS back into another class of disorders, impulse control disorders as well. And so there's sort of a lot of new interest. And of course, there's always new interest, searching for ever broader categories of indications for DBS, but that's why a lot of the ICD folks, impulse control disorder folks, are interested in DBS, subthalamic DBS in particular, because of that trade-off between speed versus accuracy in tasks.
So question, most of the STN stuff that I've seen is more focused on caudate putamen side of the story. Like, there's an STN for the nucleus accumbens, yes? Like the STN projects to nucleus accumbens?
That's a good question. I've been a lot more interested in afferents.
You mean the ventral pallidum, Yohan?
I mean,
you mean the palatal,
yes,
palatal region?
Yes,
but asterisk, in that Haynes and Haber paper, they show that areas like the sort of more ventral cortical regions, like 32, I guess it would be in Brodmann area. Also, they don't just project to the SCN; they also project down to areas like that are right near the SCN in the hypothalamus, that are closer, like associated with like the medial forebrain bundle and things. So it gets really gnarly and tricky when you get over into that space. It's definitely worth giving that paper a read and thinking about that.
So yeah, that's what I meant, ventral pallidum, because I was just curious about like what is the sort of limbic hyperdirect pathway, and what does it do? So
does it exist?
Does it exist? Exactly. Does it exist?
Yeah.
Or is it just that's a really interesting point. Or is it a hypothalamic circuit? So that's kind of cool. I have to look into that.
And it's important to mention as well that the lateral hypothalamus is more like a what's that? It's like more like a piece of a place in your mind than it is a location. Like it's not a nucleus. It's an area where tons and tons of different fibers of passage lie. So all of the area, like the locus coeruleus, the dorsal raphe, ventral tegmental area, are all coursing up through there, along with a whole bunch of like, orexin neurons, all those different things. The basal forebrain is sitting right there, which is a major cholinergic hub for the cerebral cortex. And they all kind of like intersperse with one another in that lateral hypothalamic area before projecting up to the cerebral cortex and thalamus and stuff. It's like a sort of arousal superhighway in a way. So having projections in there, it's not even clear, are they hitting into neurons that then like clamp things down? Are they just going through that area? It's like a major open question, like what the heck's even going on? But the fact that they're not going straight to the SCN, I think, is really important.
This is a question for another day, probably, but I would love to hear your modern definitions of a nucleus and what our criteria are for for that. And is it even
whatever you can get published, Clayton? Yeah.
It's some combination of genetics and larger cytoarchitectonic kind of like it can't be one or the other, because if you just use purely genetic kind of criteria, you end up kind of including like the random neuron that accidentally found its way into this other part of the because these boundaries that we draw are not that strict, right? And like occasionally somebody like Helen, my PI, she's an anatomist, right? She gets so furious about like people who will count like one stray pyramidal neuron that happens to be in some place it shouldn't be, or like, oh look, we saw projections from layer 4 outside, long distance projections from layer 4. Like what? So as soon as I heard this, I was like, so they basically found pyramidal neurons that strayed into layer 4, and she was like, yeah. And that's exactly what they were. It's like as if there's some wall on either side of layer 4 saying pyramidal neurons are not allowed. So anyway, okay.
I actually think just to kind of like put a cap on that thought, I think Clayton, your point is really, really excellent, and this is perhaps something that maybe modelers could teach neuroanatomists in a way, is to say the principle is probably more important than the actual detail in some cases. Not always, and sometimes the details carry the day, but if you think more about what could this circuit do versus not, or what would a neuron sitting in that area be able to do that it couldn't do if it was sitting somewhere else, like the principle of the anatomy, I think it can actually help you to classify it in a way that genetics or like the specific precise location may kind of miss. And so I think there's actually a really good place for like a bit of a dialogue here between the two fields, which is why it's so cool that someone like Yohan sitting in Helen's lab and having to listen to them talk about anatomy all the time must lead to fun conversations.
It's really interesting, and having to explain and even just the way that one draws diagrams. It's like I used to always draw these very simple straight line diagrams of what's connected to what, with no attention to where anything is. But now I try to like make it more look like something that is in the brain.
How much do you believe that the answer to that, or at least how important of an answer is this, the interplay between anatomy, geometry, and spatiotemporal dynamics? Because the location of something determines how long or how quickly it can respond to something and how long it takes for it to pass the signal on, right? And so if you take a network and you change the geometry, you're going to change the spatiotemporal dynamics, right?
Right. But then this is something that bothers me because I don't know how to do it in an efficient way. Right? We are hardly ever, when we make network models, how many of us include conduction delays? And do you find strong qualitative differences when you add those conduction delays? I'm very curious about this actually.
Well, I mean, a big reason why I do that is I actually am studying electrophysiological biomarkers of therapeutics. So I'm really interested in, can we use this ECOG signal to say, yes, this is therapeutic DBS or not? And I can't do that unless I can tie this pattern of recruitment and pattern of conduction to this signal at the surface of the cortex. So it really matters how long it takes for it to get there. And it really matters, the actual, the micro scale morphology of these neurons really matters, because that's what determines, does this field recruit this region of the neuron, and how long does it take that signal to get back to, to my output signal? So I model things like that, but I see a whole lot of network models that ignore things like network latencies. And I'm really curious.
Yeah, it just makes it even harder than it already is, right?
It just makes it a big nightmare.
This actually came up, Clayton, in the world of like macro scale human brain modeling, when you kind of pretend like the brain is just the cortex, and the cortex has 400 regions in it or something like that, and you try to think about, well, what if we had some principal way of linking these nodes up based on what we think the structural connectivity is? What kind of patterns will we get from the circuits if we have a sort of very sensible model of neural dynamics? And the group found that, this is Gustavo Deco's group and Vic Ursa, they found that if you incorporate a delay that's proportional to distance, like 1/r to the 4 or something like that, something sensible, and you get really different kinds of dynamics, different kinds of local patterns, different kinds of matches to data. They were actually better than if you had a model that didn't have that factor involved in it. And so, you know, in that case, they got a lot of benefit out of it. And there's another group as well, John Murray's group at Yale has done some stuff where they matched model to, I think, MEG data. And they found that if they incorporated a gradient of the model that had a timescale factor in it that mimicked the complexity of the changes across the cortical mantle. So, you know, prefrontal cortex looks a bit different than sensory cortex, let's say, then they could get a better match to the MEG data again. So I think a lot of these things can actually be helpful additions rather than harmful if you can find the right way to put them in as well. But,
you just have to make sure you don't fall into the tractography, you know, pit, which is assuming that that brain connectomics are this sort of like binary wheel model. And
yes,
it's that's not how brain connectivity works, right? And yeah, and if you think about
more like spray paint than ropes, right?
Right, exactly. And you know, I mean, it's better than no data, right? And so, you know, we use it, and you know, we want the data, but at the same time, you know, we're talking about broad cortical regions that communicate over centimeters, you know, tens of centimeters, and there are meaningful delays. And when you talk about the strengths of inputs, the latency, you know, even a really strong input, if there's a strong latency as well, then to a certain extent, it mitigates the value of that input because it's so delayed, right? A fast, weak input may end up being more important than a strong, slow input.
So what timescales are we talking about here? And how do you determine the value?
Yeah, so pyramidal tracts, for instance, I mean, I have to put this in my context, right? But, you know, for DBS, fast antidromic EP could arrive at the cortex within 3 milliseconds.
And so what would be an example of a delayed, but therefore?
Yeah, so let's imagine your pyramidal, your internal capsule neuron is, say, 10 microns in diameter. Then the antidromic latency might be 3 milliseconds, but in reality, the vast majority of pyramidal tract axons are in the neighborhood of 1 micron in diameter, and it might take 10 or more milliseconds to get back to the cortex. Okay. So you've got this sort of like skewed distribution of antidromic activity, and you know, by the time we're talking about 10 milliseconds, that could be, you know, any number of different common inputs to the cortex, right? So it gets a lot more hairy. Trying to say, this signal comes from here, like it doesn't necessarily work.
I see. So it relates to the difficulties to do with spike sorting, for instance.
Spike sorting, but more functional, I think, functional concerns. And it's even longer for prefrontal cortex. So that those numbers were for motor cortex. Now prefrontal cortex actually, you know, goes, you know, forward and up, right? And so now we're talking about even longer signals. And when you're in the neighborhood of 15 milliseconds, it could be any number of different, you know, common inputs. And so you can't really say, you know, this pathway, activation of this pathway causes this cortical, you know, network activity. It gets a lot more hairy.
Yeah, I've been thinking about whether I should include delays, because I know that in things that work more like rate models, where you don't have that many oscillatory kind of phenomena, it doesn't really do a whole lot. When you're doing like a competitive network, it's not going to be strongly affected by those kinds of delays. So this is more like the kind of proof of concept. This model does this particular thing. But when you have inhibitory feedback, then you can get it's basically an infinite dimensional dynamical system, right? In the sense, so you have all these possible things that can happen as a result of the recurrence. So what might have just been tonic activity without the conduction delay would become some sort of gamma oscillation, for instance.
You have to kind of throw out all of the basic tools of analysis network.
Yeah. So yeah, but yeah, I remember playing with conduction delays and spiking models a while ago, and I was like, oh boy, there's a lot of stuff that I don't that wasn't qualitatively qualitatively related to anything that I wanted to point to. So it like you could put it in there and then have to adjust other things to make things work. But it's interesting to the place where I would eventually like to incorporate something like this is like our lab finds a lot of differences between these sort of medial limbic kind of cortices and the laminate ones in terms of how much myelination they have. Obviously, the well-laminated areas are much more myelinated. But there are a few exceptions, like MPAL, right under behind the eye, and and another interesting thing that people have observed is, for instance, the inter-regional communication, which tends to be involve myelinated axons, can happen before lateral communication within a brain region. So you can have sort of V1 to V2 and back, while any kind of lateral communication within V1 hasn't really done much yet. So those are the places where it will become quite interesting. We sometimes when you're dealing with behavioral stuff, the reaction times are 50, 100 milliseconds or something like that. It seems like a 10 millisecond, 3 milliseconds may not matter all that much. Maybe it will when you have a few sort of stages of this kind of thing happening. So,
yeah, I just find it fascinating that, we tend to think of white matter as like this homogeneous thing. And in reality, white matter, has dramatic differences in conduction velocities, like from 8 meters a second to like over 100 meters a second. And that's in primates. We don't even know in humans. It might even be faster than that in humans, right? We just have to sort of guess. But when you're talking about those sorts of distributions of conduction velocities, sure, region A connects to region B, but there's actually this transfer function between the two regions that changes the signal dramatically.
And
I've heard. Were you about to go in the learning direction with this too? Okay.
Please,
yeah. Well, I mean, I don't have actually any citations for this, but apparently I've heard like through the grapevine that there is some plasticity in the myelination itself.
So the oligodendrocytes have synapses on them, right? So in principle, you can have activity-dependent myelination, and this makes me really happy, even though I have no idea how I would actually model this. And to my mind, it'll be like some kind of nightmare, right? Because you're trying to basically move the alignment of spikes using myelination, and obviously it'll be slow. It's not that fast, but still, it's really interesting to imagine that you could use the activity of cells to change how fast the communication happens. And when you, on its own, it's not clear why that would be useful. But if you combine that with STDP, like spike timing dependent plasticity, then you really have like a set of ways of doing synaptic plasticity where our intuitions are like, it's really hard to know how to get it to do something useful. Which is why I never really looked into it. But I think Stefan was modeling it, I think. And you can get it to stabilize. In fact, that was the first thing I asked. Would something like this be stable? And you can get it to stabilize it, like so that you're not constantly speeding things up or slowing things down.
You give me a learning rule, and we'll build a model. I'll give you a biophysical model. If you give me a learning rule.
Look back at this. Nice. Yeah. Yeah. So actually, I was going to just move on to. I didn't have time to find a decent model of the Mar Albus thing, and I didn't even look at it clearly. But do any of you remember? So it's kind of like, to my mind, learning a lookup table. And I was going to draw it out. Let me see, how do I do this? Since I'm a lefty, I find it hard to draw directly on the screen when it's vertical. But let me see one sec. Oops. It's always so weird. So basically, you have all these inputs coming in from those parallel fibers. the Purkinje cell is represented schematically. It's just this line here. So this is supposed to represent where the intersection happens. And the model just involves kind of changing the synapses on the Purkinje cell so that you can remember basically all the patterns that show up here. And David Mar and Albus, they all kind of, and Ito all had more or less the same idea based on that diagrams from that book, Eccles, Ito, but so the idea from behind this is that you might have like some pattern here, some vector, right? That's the activity coming in here. And you have some teaching signal such as that, what is it? The inferior oblique, right? So the coincidence of the teaching signal, which is presumably low dimensional, so coming in here, and activity here, and the Purkinje cell also needs to be active, I think. So it's a Hebbian kind of story. It can cause you to learn pretty specific patterns. So you can basically use these coincidences to learn patterns. But like I was saying, the fact that there's a sort of fan in makes this a little complicated. I don't quite know what it would mean to learn a pattern like that.
You mean the fan in from the granule cells
from the Purkinje cells, the output, there's a fan in.
Oh, you mean down to the deep cerebellum nuclei?
Yeah.
There's been some interesting work. I'm not super around this completely, but I have to dig up the references. There's been some work showing that it's much easier to explain the activity of Purkinje cells at a population level than it is at the individual neuron level. Okay. Such that it's not, because part of the Mar Albus model is the sort of assumption that a little bit like the Alexander DeLong and Strict model with the basal ganglia, that individual Purkinje neurons were kind of like gating a particular output and therefore allowing that output out or not. And so if you silenced one of them, then it would no longer be able to inhibit the one that you would like to do in that context or something like that. Whereas I think the sort of more modern way of thinking about it is that there's some distribution of activity patterns across all the Purkinje fibers that changes the likelihood of like individual Purkinje cell becoming active in a certain context, and through their population coordinated activity somehow gate the subset of the dentate or whatever nuclei that then go out and then have an influence. And it's a rapidly evolving signal as well. But I don't know that stuff well enough to quote proper articles or anything like that. But I'd read some really cool papers along those lines, a population level thing.
Is that part of that? Like, I don't know the terminology here, but I've heard people talk about this, like a manifold, population manifold. I don't know.
It would play into that. So most of the people that talk about manifolds in my world are talking about just cortical, like sort of, brain imaging patterns. And then there's also some cool stuff where like Ilya Feet had looked at the manifold of like like head direction cells. And so there are definitely people that think about that at that level too. But I haven't seen too many people tackle cerebellar circuits and corticocerebellar circuits with manifold things. The one group that has done some really cool stuff is Adam Hampman's group, who have shown they have this really cool set of work. Britton Salbry was the first author. As a Nature paper I could dig up, but they had this kind of funny experiment where there was a mouse in the dark with a little camera stuck on its head on a ball, and there was like this little kind of like wheel that could go around that had pellets every now and again in a bunch of little holes. And what would happen was the pellet would the thing would click around, and then the mouse would have to reach out to grab the pellet, which it can then eat. And they could mess optogenetically with different parts of the corticocerebellar circuit to get it to like miss and like reach out and then not grab it, or after they grab it, drop it, and all these different things. And they used the population. They did some recording, I think, in I can't remember if it electrophysiology or calcium imaging in parts of the brain, and could show that there were kind of low dimensional patterns that stimulating parts of the cerebellum or the thalamus could like knock the neural activity off of its manifold into a new kind of part of the space. And if it recovered back to the manifold, then the animal could like, or it would be also like dropping the pellet but then grabbing it again. So there's all this super cool stuff out there that's starting to ask these questions and messing with things. Yeah, super cool.
So if we were to like sort of just expand on this circuit, right, and just flesh it out, the dentate is excitatory or inhibitory?
It's excitatory to the thalamus,
okay,
and inhibitory to the inferior olive. So it'd do something, yeah, so like that, and then it would have a projection back here.
So it has 2 different types of cells?
From my understanding, and I don't know this stuff in enough detail, it's the same groups of cells with different projections, but I don't quote me on that.
Okay, but they're glutamatergic, yes?
So I believe, I know that for sure that this signal here is glutamatergic. And I know that people talk about dentate nuclei projecting to the inferior olive, and they talk about it as an efference copy. And if we're assuming for parsimony that these are all glutamatergic cells, let's assume that there's an inhibitory neuron here that's inhibiting the olive for now. I don't quote me on any of these, on the specific details, because I just don't know.
And so
this is the efference copy here when they talk about it, right? The dentate nuclei said it's decided to do something, and then it tells the olive to shut up. That's how I always thought about it.
Right. It's like almost like saying that, you know, something has been done already or something, like check.
But the I don't suppose that the olive is rebound bursting, is it?
Interesting. Good question.
That's a really good question.
Wow.
The olive is an excitatory nucleus. I think that a lot of these patterns that you remember we talked about that kind of complex spike. So that it turns out that Purkinje cells can fire in 2 modes: simple spikes, which kind of just look like spiky, and complex spikes, which are, it's effectively just another way of saying burst. And the story is that the difference between these two is when the olive comes in, wraps around the dendrites of one of these cells and releases a bunch of calcium. So it'll probably be some kind of metabotropic glutamate receptor, I'm guessing, that comes in and then that changes the LTP and LTD as well. So you get learning as well as a change in the function of the cells. Yes, so they're excitatory rather than inhibitory, right? So it wouldn't be like the burst, we talked about last week, which are from inhibitory cells, right? Like the pallidum or something.
Right. I think Bob is just wondering if by virtue of having, say, a low threshold calcium channel, you can cause it to, you know,
got it.
Meaning, does the inhibition itself potentially cause more spiking? Yeah.
Right.
Got it. Okay.
So
I don't know that answer to that one.
The thalamus, the parts so we saw that in that paper they mentioned one of the intralaminar nuclei, but the ones that you said CMPF, right? Is pretty.
Theirs wasn't CMPF. Theirs was.
They were talking about
nucleus. Yeah. Let's just call it intralaminar nuclei.
Yeah. I think they said CL or something, which is one of the intralaminar nuclei.
Mm-hmm.
And so we have, and these do not project to prefrontal cortex, right?
I think they're more likely to project to the striatum, but a lot of this data you got to be careful because it comes from rodents and we don't know what happens in humans. I guess it depends what you're seeing. Yeah.
Because M deep is what projects to most of the prefrontal areas.
Yeah.
So these go to striatum, you're saying?
That's the story. Yeah.
So does this.
But MD is in this category here for primates and humans. It receives deep cerebellar nuclear output.
Okay. See, this is good because this is what I wanted to get everybody to start speculating about, right? Which is, so I'm going to just insert MD among these.
I actually know. Hold on. If you give me 2 seconds, I'll be able to find the name of the subgroup of nuclei within the MD that they project to, because I looked this up for a recent paper.
So I'm just going to make this now a generic thalamic node for now, and say It's whatever receives this. Okay. And so now we have our, let's do it this way. We have TRN doing what it does.
It's the parvocellular medial dorsal nucleus, if anyone cares, which nobody does. So I don't know why I bothered.
Oh, like part of MD is what you're saying?
Yeah. MD has all different kinds of cells in it, like a lot of regions, and they have different predilections towards receiving input from different other structures. Yeah.
Right. So
but the parvocellular cells that receive the cerebellar input are the ones that project to DLPFC, like 9 and 46 in humans.
So let's just do this then. This is your matrix type projection, which is going to the upper layers mainly. And the key idea is that it goes to upper layers and it's kind of widespread. It also goes a little bit to 6. That's matrix. And then you have core cells, not necessarily in this kind of relationship with TRN, and these tend to go to 4, upper part of, lower part of 3, upper part of 5, which is 5 is where you have the big pyramidal neurons. Pyramidal neurons here and here as well.
Which are the same ones that project down to the pontine nuclei, which are the main input here.
Which ones? Well. 5 or 6?
The 5.
Okay.
The PT type. So the BET cells in motor cortex, the 5Bs.
Right, these big ones go there. So yeah, and in limbic cortices, the medial prefrontal, posterior orbital frontal, etc., it's not always easy to distinguish 5 and 6. So you have to kind of be a little careful about these things for these like medial and orbital areas. And the same issue will come up when you're talking about which of them project to TRN and thalamus. So this is the standard way that this is drawn. And we don't really know.
I read in Ted Jones's book that you don't see the same. So like for the cells that contact the matrix and the matrix on the way out, you don't get the same TRN involvement, but they're affected by the TRN. That's according to Ted Jones, 20 years ago. So I don't know
collateral. You mean this?
The story from Ted Jones's work was cells that end up contacting the core and the core cells that end up leaving both perturb the TRN.
Yes.
But the cells that contact the matrix and the matrix cells on the way back out do not. Yeah. But the matrix cells still suffer from the core-induced increase in TRN inhibition.
So this exists.
That was the story, but that's from a long time ago.
Right. Yeah, because this is hard to study basically.
Yeah.
And apparently it's really difficult to inject TRN cells, and you can kind of maybe fill them in mice, but in primates apparently it's super hard to. So there's a lot of things that are just very difficult to know about this circuit. So getting back to the speculation, what does this mean for cognition? If we're talking about a corticothalamic loop, that is not a motor loop. Or even if it is a motor loop, what exactly does it mean, right? If we're saying that there's all this fine timing or whatever kind of representations that are coming out of the cerebellum, what does the rest of the brain do with that? Like, when does any of that reach muscle fibers, for instance? Not via here, right?
Well, so some of the DNs that go to the magnocellular red nucleus.
Okay.
Sorry, I've lost the ability to project. The magnocellular, these are the ones that project down to the spinal cord that control neck movements.
Okay.
And upper limb movements. So like the cervical spinal cord.
Okay.
But they're systematically diminished in humans compared to non-human primates.
Right.
But the parvocellular red nucleus becomes massive So it's like the dentate nucleus and the medial dorsal nucleus and the pontine fibers. It becomes huge, and this is where it gets really interesting. It projects into the inferior olive.
Oh boy.
So I don't know necessarily if you want to include that. That's a story for another day. But it's
no,
very interesting
awesome.
to think about what that's conveying.
It's good to lay out as much as we do know,
Yeah. And there's some tantalizing anatomical stories that suggest that different populations project to parvocellular red nucleus compared to the pontine nuclei. But when I chatted to Gordon Shepherd about that, and he thinks that some of the controversy comes from the fact that the old labels were somewhat indistinct compared to the modern genetic labels, where they can really talk about specific populations of neurons and projections. Right. But again, the big problem is that we just don't have great anatomical stories and data about specific subpopulations in, say, humans or non-human primates for a lot of these areas. So let's say you wanted to make a conclusion from the Allen Brain Atlas or something. Well, it might be in mice that the vast majority of magnocellular cells, the ones that are really motoric, are receiving inputs from these same layer 5 pyramidal neurons. But the ones that are the more involved in driving the olive, which don't really exist all that extensively. I mean, they're there, but they're not as elaborate in rodents. Maybe they get something different, but you just don't have enough tracings to find it properly. And so it's a really murky area that we just don't have enough evidence for, I would say, yet.
So you've drawn superior, that's SC, you mean superior colliculus, right?
Oh, sorry, spinal cord.
Spinal cord, okay. Good.
Apologies.
Okay.
Yeah. The cervical spinal cord.
I'm a little confused. Spinal cord. So in principle, for the pure motor effects of cerebellum, that can happen just via the output directly. So this is where the question, the cognitive question comes in. If we assume that the cerebellum is providing a benefit to, like, what is it? Cognitive dysmetria is a phenomenon that people talk about, right? So then we have to talk about this kind of whatever highly learned, potentially overtrained kind of information, doing something to our corticothalamic lobe in principle, right? And okay, also to the effect on the basal ganglia, which is also via thalamus anyway.
But I think it definitely is important to mention that while Bass is right that there is a blend of all these things, the evidence that we have suggests that cortical outputs to the thalamus are overwhelmingly likely to contact core-like nuclei. The VL, where it projects, is almost exclusively core with a little bit of matrix, whereas the VA, where the basal ganglia hits, is almost exclusively matrix, a little bit of core. So there could be some contacts between those, or like a non-specific projection patterns. But that CL that they mentioned in the paper is like a tiny aspect of the projections. I've chatted to Laszlo X80 about this, who's like a big thalamic guru, and so the vast majority of the cells, like for example, if you look at staining, Laszlo has a really lovely paper where they look at staining of GLUT1, GLUT2, and GABA. So GLUT1 is a cortical origin glutamatergic cell. GLUT2 is a subcortical origin glutamatergic cell, and GABA is obviously a GABAergic cell. The VA is just chock full of GLUT2 with very, very little GABA. VA is the opposite. It's chock full of GABA with tiny little amounts of GLUT2. And so the argument is that I think we should, if we're thinking about like what is the impact of the cerebellum on cortical processing, it would be a bit silly to think of the kind of the minimal example, but rather better to think about what the benefits of the majority of the connections are. And then once you have that done, you can think, well, what would be the benefit of having these other signals, to recruit the striatum or to hit the LC or the VTA or something like that, because maybe they're important things to be able to condition on. But for the most part, I think it's the exception rather than the rule.
Yeah. No about that. But yeah. So, but my question was just, even when you take that into account, what would a signal like that, which is excitatory, offer in the case of like one of the papers I saw recently was about some sort of mathematical like mental math.
Oh, is this McDougall's one with the working
Yeah, the one
memory?
that I shared like a couple of days ago.
Yeah.
So that's the thing, right? So from the perspective of like what exactly is this particular cortical thalamic loop doing with the cerebellar information?
So I mean, this is where you're right. We are in wildly speculative territory, right? But this is where coming back to that kind of idea about the origin of the cerebellum, and it actually kind of comes back to a question from before. I can't remember if it was you, Rob, that was asking about this, or Clayton before he left, about the fact that the cerebellar thalamic targets look a little bit like what we think of the primary sensory relay looking like. Is that the cerebellum, even though it gets called a motor structure and it clearly helps motor output, is actually quite clearly a sensory structure, in that the way that it originally began was to cancel out motor actions, so you had a really better, more sort of precise understanding of the sensory inputs that had changed relative to what you'd done, right? Yeah. As like what John Montgomery calls an adaptive filter. And so if you think about it like that, then the way I think about it is that the cerebellum is sort of improving the frontal cortex's sensory information, but the sensory information is no longer whether or not you heard a sound or saw a particular item in your periphery, but rather whether or not the cognitive, like in the abstract sense, it's sharpening your cognitive, ongoing patterns such that they can do more with them. They can act more quickly. They can fill in the gaps more effectively. They can anticipate the next step in a sequence more quickly, so that you can build more complex abstract concepts in your sort of like cognitive repertoire. So you know how hard I am trying not to say the word representation right now, Yohan. I hope you appreciate that.
But anyway, I was going in the opposite direction, because I was saying, suppose, like, again, this is a case of like, let's force a sort of very simplistic analogy and see how far we can take it, right? So if we're going to say that our model is this, the sensory compensation idea, right? Let's just stick with that for now. Might not be the whole story, but let's just suppose that animals, primates in their trees, one thing they're really particularly good at is knowing, eventually learning the consequences of their actions, and dealing with and factoring in at least the first order consequences of those actions, in order to not be surprised by the fact that the branch that you're standing on is waving, for instance. What would that mean for, so suppose that we again say that thinking, planning, whatever is a type of movement, then what's the analog of the sensory consequences of self-movement? That's what I'm saying. So what I'm saying is, what is it that you might consider that, like, I'm planning, so I'm manipulating some entity. It's completely hand wavy, right? Some sort of manipulation is happening that we're treating as analogous to movement, and that will have some consequences internally rather than in the world, right? So internally, by virtue of me thinking about an apple, all kinds of other associations turn up. And so could we then say that having loaded up certain types of things, like some winner in a competition within the cognitive domain, then you need to kind of anticipate what will come as a result of thinking about that and compensate for it. Let's say maybe there's a bunch of irrelevant things that might come up when you think about the word apple. So that given the task that you're in, you need to anticipate, well, I really like this particular kind of thing, but I shouldn't reach for it because this is not the time for it. So something like that is, the way to really take something that we think we understand that's very physical and see if we can avoid the word representation via this type of thinking. So does that make any sense? See what I just said?
No, it does. I think, like, in a lot of ways, it comes down to that kind of Sisekian viewpoint that you came at before, which is sort of like, it's not really decision making that's distributed everywhere. It's sort of an arbitration process, and the thing that's being arbitrated over can be really concrete, like move this muscle fiber group and inhibit that one so that you ultimately move your arm, or it can be really abstract, like change your goal state so that instead of looking for food, you now look for shelter or something like that. And as long as you allow for that sort of abstraction, the potential options that open up between, I would like a set of different things to happen and I can move in these particular ways, is astronomical. And particularly as you start to expand out the prefrontal cortex and the parietal lobe, now you've just got like billions and trillions of different possible things you could do. While you may have a kind of inbuilt almost like implicit response to an apple to think about eating it, you could also throw the apple, or you could crack it in two and use it as a stamp like we used to do in kindergarten, or you could, if you were so inclined, you could core it out and turn it into a bong or something like that. Like there's a billion different things that you could do with an apple, and they all depend on the goal state, and they all depend on some constraint of like what the apple's internal makeup is and your way of interacting with it, the tools you have around you, but also your body. But I think that intermediate zone is going to be way better off if, based on the particular goal you have and the particular options that you have for executing that goal, you can anticipate what ought to happen if you take that step. So if I go to crack the apple, I know that it'll probably break. But if I hit it too hard, it'll probably smush. Whereas if I was hitting something like a brick, it's unlikely to smush because I don't have anything strong enough, but it could break if I hit it the right way, right? Like having that anticipatory knowledge of the way that you can interact with the world can change fundamentally how you can plan ahead and anticipate. And the better you can do that, the quicker you can get to the end point, the solution.
Right. So if we like look at a fuzzy level, there's like, if you're thinking about something vaguely like planning, we have this atemporal anatomical circuit, and we don't know, if there's some order in which there's an operation. But suppose that we just choose to say that there's some, let's say, a tentative plan that starts out here. Something that is sort of like coming in from cortex, and then we're going to do a cerebellar loop. We're going to access something that has been previously learned, let's say. Now, the thing that we can potentially talk about is it's excitatory, right? Like whatever has been pulled out of there is excitatory here in thalamus. What does that mean for like maintenance of that particular thing? Because like if it were inhibitory, you could say that you're for canceling out the consequences or something. Yeah? What I'm saying is we're not exactly canceling out, right? Or at least if we want to say that we're doing something like this, we have to kind of provide some additional infrastructure for that. So, for instance,
so when I think of canceling out the consequences, like there's that, I don't know whose phrase it is originally, but the sensory motor anticipation, or like anticipating the consequences, the sensory consequences of motor action. That's the term. I don't necessarily think about it as canceling out something, but rather filling in the likely next step such that you can kind of trust that it will be there and act upon it quicker than if you didn't anticipate it. So that while it ultimately leads to a anticipation, it doesn't necessarily mean a cancellation. That's how I think about it anyway.
Instead, it's something like priming the next step. It's predictive coding, whatever you want to call it.
Yeah, that's how I think about it.
Okay.
That may not be the right way to think about it. But.
Oh, this actually helps a lot, actually, because this makes a whole lot of sense from the perspective of an excitatory signal. Particularly if you're not simply going back to where to a corticothalamic loop that you originated in. So here you can imagine a kind of daisy chaining, right? So you have something in cortex, something in thalamus, and so this goes to, I'm just going to short circuit that, cerebellum and whatever, and back, right? But suppose that it's not going back here, but here. Now we already have an anticipation mechanism, right? Which is that other inputs potentially were, or even from the cerebellum, let's say, are saying, well, this is what's happening now. This is active or strongly active, let's say. And because of potential inhibitory interactions, there's some amount of competition happening to locally, or and something like this loop here could be like, well, prime this particular patch, because
you're drawing slightly off the screen, Johan. I don't know if you can move it side to side.
Yeah.
Yeah, I mean, I definitely, I think this dynamic loop way to think about it is absolutely the way we should be thinking about it, right? Like every time you take any kind of action in the world, you automatically have an implicit set of assumptions of what ought to happen next. And sometimes they're very well developed. Like I know what it would feel like when I put my leg out to move and walk on steady ground. And sometimes they're terribly well developed. I don't know what it's going to feel like to float around in space. I've never been in zero gravity before. Right. And so but I still will have some set of anticipations. I think it will be like this, and I'll be woefully incorrect. So I'll get all kinds of crazy prediction errors around my system for trying to anticipate in the way that I was doing. And then my job is to update my new model of you're in a new context now that you didn't really realize before. None of the old stuff's working. You've got to try new stuff. You've got to try to learn the new rules.
Sorry, I might have missed. I might have missed some of this because I was trying to see if there was rebound bursting in the inferior olive, and it doesn't seem like it, but I'm not sure. But one of the things that's interesting, at least in terms of like motor control, motor planning, like these kinds of things, like there's what you're doing right now, and then there's like a kind of tree of stuff that you can do the next step, right? Is that where you guys were kind of going with this? Or?
Yes, exactly. Yes.
Okay.
Yeah.
It's like the.
And I think the cerebellum will increase the dimensionality of the tree.
Yeah.
But in such a way that it's going to lead you to quick and adaptive solutions. So assuming let's say you're an expert, your tree will be high dimensional, but will be collapsed to the set of dimensions that were most effective in allowing you to complete that task in the hundreds and thousands of times that you've done it before. Picking up a bow and arrow, you know where your elbow should go, you know how it should feel when you pull back on the string, you know where your arm should be, you know how to take care of the wind. If I'm a novice trying that, I try my model on it. I've got a high dimensional output, but it's like a mess, right? I don't know where to put any of these things. I've put my elbow up higher before, but it's when I was writing on, a piece of paper that was up here, and I've got, my hand steadying something, but it was when I was steadying a steering wheel. Like I've got some preconceived notion of what those movements ought to lead to, but they're absolutely not fit to the context. Whereas an expert is absolutely fit to that context. So they can get quickly to that anticipation such that they can then have a much, much more effective output. So it's tricky, right? Because the expert will actually have what looks like a low dimensional response because they've learned how to do it so effectively with so little waste. Like Steph Curry shooting a jump shot has so little waste to it. Whereas if you watch me shoot 1,000 jump shots, mine will all look different. Some of them will be over here, some will be over there, whatever. But Steph has learned this is the particular motion in this timing that leads the ball to go in the hoop again and again and again and again. The cerebellum can buy you that specificity. So I don't know how to translate that to like mathematical notation, but I think the function, it makes a lot of sense from the circuitry.
It's sort of what I sort of did. It's like there's a lot of things that could happen, and with training you kind of narrow it down. So with the dentate, if it's excitatory, where does it get excitatory input from? Because the Purkinje cells are inhibiting the dentate, right?
Yes, but this is where I think the population encoding thing would make a little bit more sense, right? If you can learn that a particular context and a particular set of opportunities mean that it would be great if this subset of thalamic channels could be open, which in terms would then prime this set of pyramidal neurons, then as a whole, the Purkinje cells can be like, okay, cool, you 14 shut up and everyone else be really active right now. And that'll mean that you can get that particular sequence out. But rather than it being it'll be those 14 for a particular tiny window of time, and then the next 25, and then the next 32, and then the next 5, and then the next 16. And you learn that particular sequence that it'll then, it's almost like, I almost imagine like a player piano where it can like roll over the little dots, which can then make the piano keys play. It can't play anything else, right? It's not capable of adjusting. If the piano was out of tune, it couldn't think, oh, I'll translate everything up half of a key and then play everything on that, and I can do that to make the sound better. It can't. It's absolutely stuck doing the pattern that it has learnt will lead to the optimal output. Right. And it can do it really quickly. Yeah.
So, but does that mean that the dentate nucleus is like perpetually firing kind, like an intrinsically firing kind of a cell? Or?
Yeah, it is. Yeah.
Okay, good. Okay. Yeah.
So is the so are the Purkinje cells. Yeah.
Okay. So cool. That's interesting. So it's almost like if you have a complete failure to inhibit the dentate, you have some kind of really, really weird experience of everything is possible, kind of, or all sorts of things get predicted all at once.
Maybe that's how chorea happens, right? Like those like hemibolismic movements.
Whoa.
That's an interesting thought. I hadn't thought about that before.
Really interesting thought.
Yeah, I have to think about that some more.
Well, I don't have anything else to go on with this analogy, but if anyone wants me to draw out more stuff, or if anyone has anything else to say about this, thoughts?
It's a tricky area to, so one of the things that frustrates me about the cerebellar circuitry is that it's very hard to think of a way to reduce it, coarse grain it, and maintain the things that it probably is doing quite well. So one of the things that makes it really interesting, right, is that the granule cells are there's just billions of them. They're half the neurons in the brain. Right. So in a way, they're kind of like augmenting the sort of dimensionality of the signal. That's at least one of the theories for what they're doing to the pontine nuclei. But you can't really kind of like summarize that down particularly well in like, you know, a little neuron, the way you kind of comfortably say, oh, well, let's pretend this cortical area is just one neuron and we'll just sort of like leave it be. It's much harder to do that with the cerebellum. You got to think really carefully, I think, about how to do that.
This is true.
Collapsing.
So like, in fact, that reminds me of how people talk about in the hippocampus, how people talk about the dentate. So the dentate has a lot more cells than the entorhinal. So if you have cells on the entorhinal, I'm just drawing in the dots for now, but and then the dentate gyrus, not to be confused with dentate nucleus of the cerebellum. Now we're talking hippocampus. Is the idea is that you get a kind of sparsification. So cells that are patterns that are similar within the entorhinal cortex. Let's say I'm going to use Venn diagrams to talk about patterns being similar. When they project to this larger group of cells, I'll give myself much space. This larger group in dentate, the idea is that the overlap is relatively much smaller. So they call this, they assume that this is crucial for pattern separation. And this is really kind of cool in the context of episodic learning, because you want to be able, like most people are able to remember that today is different from yesterday, even if relatively similar things happen today and yesterday. So in a state space of things that happened, you might find, you know, day 1 and day 2 are in some sense similar. So you kind of want to project to this larger space. It might have more dimensions inside, or a lot of redundant dimensions, so that in this other space, day 1 and day 2 are somehow much further away from each other. Oh, I've done that again. So the idea for the cerebellum would be something like that you're using one way to think about fine control is that you need to maintain fine distinction. And another way to think about fine distinctions is that you're moving up into a space where you can really reliably talk about small differences. So there's some analogy to be made between what the cerebellum is up to and this EC to dentate kind of story, which I agree is not.
Have you seen the paper I just sent, Yohan? I can't remember if we've chatted about this one before. Angus Silver and Alex Keiko Gajic, I don't know how to pronounce it properly, but they make exactly this analogy. They say that the the structure of the cerebellum and the structure of the hippocampus is actually extremely similar in terms of the fact that they do a dimensionality expansion before a collapse.
Yeah,
and
yeah.
it's actually similar to the mushroom body of a Drosophila, which is apparently important for it to do a whole bunch of adaptive things as well.
Cool.
So I like that idea.
I think it's very wide. I think, John Listman, if you read some of his stuff, he makes this point. And Dan Bullock actually made this analogy recently when I was talking to him, which is why
cool.
it was in my mind. So it's probably out
I want
there
to
in
meet
the
Dan.
literature. So this is the and in fact, that makes a nice bridge with the machine learning world. I'm not going to elaborate too much on this, but there's this whole with support vector machines. I'm going to make a mush of it unless cortex saves me. But basically, there's this idea that if you're trying to separate data points, so like in, let's say, the initial space that you're in, you've got some data points, and they're kind of all a little bit mixed, right? And machine learning classification is trying to separate them, right? To come up with a separating hyperplane to categories. So the idea is that if you can move to a different set of dimensions, like 4 dimensions, or much higher dimensions typically, and there the separation will always be, you'll always be able to find a linear separation. Which is a super cool
I love this idea.
Of I think Vladimir Vatnik is the one who showed this. So a lot of people who are who span that kind of neuroscience ML world like to use this type of idea as a possible way to talk about, particularly hippocampus, maybe cerebellum also. It could help, but in a kind of more temporal and dynamic way. And so that's one idea. The other one is actually compressed sensing, but I don't understand that well enough to elaborate. But there have been people who claim that this circuit does compressed sensing. There's a review paper in, I think, Frontiers talking about the hippocampus doing compressed sensing. So there's a couple of different stories. Yeah.
Are you familiar with Dan Schachter's work? Do you know that name?
Yes, but I haven't looked at any of it lately. So.
Yeah, the reason I bring it up is that he has this nice kind of categorization of memory function into 2 broad categories of declarative and procedural memory.
Oh yeah. And
it seems to me like declarative memory in the hippocampus with this pattern separation and procedural memory in the cerebellum with this pattern separation would be a really interesting conceptual link to make.
Yeah,
I mean, it seems like you could make the hypothesis fairly clearly that they should relate to one another, and then it becomes really interesting to think, how do the 2 structures interact and what are their connections? And that's an area that I have on my backlist to sit and think about more. I haven't done enough work there yet.
Yep. Yeah. Yeah, that makes a lot of sense. So procedural is the cerebellum. And
exactly. It's just it's anticipating the movements that you should make, whether that's a motor movement or a conceptual cognitive movement. Right, and then declarative is this thing happened here in this context, but not some other context. Like I've seen your face a number of times on Zoom, but it's today rather than some other time that I saw your face on Zoom. So it's sort of like, they're just different ways of making sure that you're very precisely acting in a context specific way.
Yeah. So just as a challenge, because I have no idea how to think about it now, I want to ask, what does this look like in a dynamical systems framework? In the language of attractors and things like that, because it would be cool to then map that onto the thalamic loop story. What does it mean to so is it something like as simple as initially starting with this broad fuzzy attractor landscape space and then becoming having these sort of sharp and distinct attractors within a broader attractor. So your BG kind of highlights a bunch of stuff in cortex that's like, okay, these are the plausible things you might be doing, and then the cerebellum kind of says, well, these are the things that are very likely to happen, this, and this.
So let's say, and then if the ball falls into here versus if the ball falls into here, this will then cause the circuit to change in such a way that the ball will kind of like rapidly roll around, leading you to outcome A. And this is the way this will roll around to rapidly make you come to outcome B. And this is where the kind of complex spatiotemporal nature of the outputs matters deeply. Whereas if you don't have any like way of thinking about what ought to do next in a purely basal ganglia mode, you would just kind of like roll around randomly, hopefully finding the solution. And then if you did, try to make sure that the next time when you came back, the landscape kind of sort of took advantage of that and didn't let you roll to the places that weren't adaptive.
Yeah.
But the language to describe attractors that evolve over time in a way that's contingent on the location of the landscape isn't something that I have come across yet.
Yeah, it's basically like Waddington, right?
Yeah, no, honestly, the Waddington analogy is the best one that I could think of. But even he talked about being at an equipotential state and then different genes kicking you into a direction that ultimately led you to an attractor that was present the whole time. You just didn't have access to it, as opposed to the wherever you are on the landscape now changes where you could go to next.
Right.
So there's an extra variable in there that makes it real tricky.
So if this really does kind of look like a tree, then I would imagine that we're probably looking at like links between saddle points. Like that's like that seems to be like the only way that it can be.
Yeah.
Makes
a lot of sense.
The saddle will have different widths and heights to it, depending on how certain you are that this is ought to what to come next, versus if it's like a really uncertain place that you could go either way. Yeah.
It's kind of hard to draw.
What variables, Yohan, would change the sort of the steepness of, like, let's say you're coming at it from this direction and you can either roll down that way or right. The flatter that this part is should tell you the more options there are for different types of saddles, right? And then, but the steeper it is, the more you're kind of constrained into the particular option that you're going to try. And then the steepness of the mountain going down the other side is how quickly you evolve into the next step in the sequence, versus the flatness is how like that. They'd be like, if I know what's going to come next when I reach my arm out, then I just go straight down to it. But if I'm in, zero gravity, I'm not quite sure what will happen. I kind of tentatively move in that direction. Sorry, my drawing there is awful, but
no, I was struggling to do this too. So yeah, I mean, it's like, but yeah, we're talking about bifurcations.
I'm thinking like
that where you're like, so the attractor becomes a purely hypothetical entity that we talk about for like slices of time in the brain that don't exist. So we say that, well, right now it's going to be trying to be here, but as soon as it lands there will be like some sort of parameter change.
Yeah.
This is why I actually kept making this point about how.
Well, this is the time delay part, though, right? Maybe that's the other way around. that is the wrong direction in time.
Like the spiking delays will do something which will fit into this general story, but like the idea being that if you had an attractor that eventually kind of stops being one, and then you have another one somewhere else, we can't frame this using a single stable dynamical like phase space, right? We want to say, well, initially the ball is rolling down this way, and then as soon as it gets here, something behind the scenes, which you can't actually draw on this graph, breaks this.
We need to all put Oculuses on right now and start messing around in 3D, right?
Well, I mean, it has to happen like slightly before it gets there, right?
Ah, nice.
It's anticipatory. Yeah. I like that.
Yes, 100%. And you know why that's really cool? Because it'll explain things like jumping the gun, right? Literally in the case of
it's one type of impulsivity, right? You thought that the context was X, but it was Y, and you acted with the X context in mind.
You're supposed to only do a particular thing after having reached here and like doing a check before rolling off. But then, like, imagine if you have that kind of mechanism where something's going to happen, which kind of affects the attractor landscape. I don't know how to draw this, but if there's noise, so imagine there's like thermal noise, right? That happens early, so that whatever it is that was supposed to happen to the rest of the brain as a result of being in this particular point hasn't really happened yet, but the system is like proceeded. That's yeah, I like it. I mean, definitely, it has to happen early. It's also that's the way to, like, because this is speculation piled on top of speculation, but the attractors that you set up as possible things that will happen can then, like a tree, like you said, at least weakly excite the subsequent things. Like humans are particularly good at thinking several steps ahead, knowing full well that some of those steps will never necessarily manifest themselves. So we know.
Like, you can also be dead wrong, Yohan. Right? You can judge someone too early, or you can think that something will happen and it doesn't, and it ruins your day.
Right. So it's not about the accuracy so much as we can sort of palpably feel that we do this. And with, as you said, like as with the case of the musicians or the extremely sort of top of their game kind of sports people and stuff like that, they are really anticipating a lot. And presumably that involves the ability to like have this space of stuff that is locally possible, and then quickly to wipe that out and then get to the next step. Because I think a lot of us, maybe we don't necessarily reset that well. So like, it's like sometimes when I play a game, something that's no longer possible still gets stuck in my head. So I wish I, like you kind of, like all those regret thoughts, like if only this had happened, then I would have done X, Y, and Z. Even in the context of a game, sometimes that happens, right? And that's a complete waste of time from the perspective of actually doing what's in the present.
Unless you play that game again and a similar situation arises, and you that time, instead of anticipating what you originally did, you can act accordingly and do it better, right? So this is why watching sports with this in mind is really fun, because the anticipations are not just something that the individual expert has, but they actually take advantage of the anticipation in their opponent. So if you're doing a crossover in basketball, you step one direction, hopefully that will cause the defender to anticipate you're driving that way, and then you cut back against them. And it's this battle of anticipatory steps. And then you can see the really brilliant players thinking multiple steps ahead, and they'll think, okay, I'm going to make them think I'm doing a crossover, but really I'm trying to pass the ball over their shoulder, but I need to get them to move in one direction. And actually in the Super Bowl, there was a beautiful example of this. Matt Stafford, late in the game, driving to try to win the game against the Bengals, right? He is watching the defenders in front of him, and he desperately wants to throw it to a guy who's going to break open behind another receiver. But he knows that there's a player there that is trying to play him off, right? So he stares at the underneath guy with all of his whole body, his whole energy, is like everything's going at this guy. And then he throws a no-look pass over the dude's head. And he absolutely was messing with his anticipation, right? He's saying, I can predict the future better than you. And he did. And it's so much fun to think about this stuff, right? It's like these crazy levels of anticipation on top of each other. It's awesome fun.
Cool.
I shouldn't have used a sports analogy, Yohan. I'm sorry.
No,
Kartik's not here to defend me.
I'm perfectly happy to listen to sports analogies. So okay, that was great. Actually, I was expecting to not be able to go very far with that line of thinking, but that was awesome, actually. So cool. Any other comments?
So look, I have to go in 5 minutes, but
this
is so much fun to think about. And I think, just to put something concrete on this, it would be really fun to think about whether there are ways to either sketch out in more detail some of the kind of like dynamical consequences of this way of thinking. Like in the 2020 Progneuro paper, I kind of like waved my hands at a lot of this stuff, but I think there's much more sophisticated stuff to say about, kind of like your point, Rob, about you need the point to come before the actual information comes in. It's an anticipatory signal, and that has interesting ramifications for timescales and things of that nature. So trying to put some concrete underneath this would be really fun. Maybe that's not for this forum, it's more a target thing, but I'm very curious to think more about how to even just frame this in the right dynamical language, so we can, because I think that's a step before modeling it. Just
to plant the idea, because you never know what will like I really think that reframing something, even though it doesn't necessarily add a whole lot of new information, can sometimes just like give you a different perspective. So anyway, I'm going to stop the recording.